Hello everyone, welcome to a new session on the applications of bioinformatics. In this session, we will study about the various applications of bioinformatics in basic research of biomedicine, microbiology and agriculture. Bioinformatics is the use of information technology in biotechnology for the data storage, data warehousing and analyzing the DNA sequences. In bioinformatics, knowledge of many branches are required like biology, mathematics, computer science, laws of physics and chemistry and a sound knowledge of information technology to analyze data. Bioinformatics is an interdisciplinary field that develops and improves upon methods for storing, retrieving, organizing and analyzing biological data. A major activity in bioinformatics is to develop software tools to generate useful data. Bioinformatics has become an important part of many areas of biology. In experimental molecular biology, bioinformatic techniques such as image and signal processing allow extraction of useful results from large amounts of raw data. In the field of genetics and genomics, it aids in sequencing and annotating genomes and their observed mutations. It plays a role in the text mining of biological literature and the development of biological and gene ontologies to organize and query biological data. It also plays a role in the analysis of gene and protein expression and regulation. Bioinformatic tools aid in the comparison of genetic and genomic data and more generally in the understanding of evolutionary aspects of molecular biology. At a more integrative level, it helps analyze and catalog the biological pathways and networks that are an important part of systems biology. In structural biology, it aids in the simulation and modeling of DNA, RNA and protein structures as well as molecular interactions. Bioinformatics research can be classified under three major approaches. 1. Analysis based upon the available experimental wet lab data. 2. Use of mathematical modeling to derive new information. And 3. An integrated approach that integrates search techniques with mathematical modeling. Current research in bioinformatics can be classified into 1. Genomics, sequencing and comparative study of genomes to identify gene and genome functionality. 2. Proteomics, identification and characterization of protein related properties and reconstruction of metabolic and regulatory pathways. 3. Cell visualization and simulation to study and model cell behavior. And finally, 4. Applications to the development of drugs and antimicrobial agents. Role of bioinformatics and its implications in various biomedical and related fields. In this particular figure, you can see the overview of various applications of bioinformatics. The following are some of the important applications of bioinformatics. They are microbial genome applications. In this figure, you can see the role of bioinformatics in microbial genomics. The revolutionary growth in the computation speed and memory storage capability has fueled a new era in the analysis of biological data. Hundreds of microbial genomes and many eukaryotic genomes, including a cleaner draft of human genome, have been sequenced, raising the expectation of better control of microorganisms. The availability of genomic and proteomics data and improved bioinformatics and biochemical tools has raised the expectation of the humanity to be able to control the genetics by manipulating the existing microbes. In the last decade, bioinformatics has been used for the microbial biotechnology in many ways. Computationally analyzing the wet lab data, genome sequencing, identification of protein coding segments and genome comparison to identify the gene function, the development of genomic and proteomics databases and inference of phenotypes, or example, higher level functions from genotypes or gene level functions. The major contribution of the bioinformatics in genome sequencing has been in number 1, development of automated sequencing techniques that integrate the PCR or bacterial artificial chromosome based amplification, 2D gel electrophoresis and automated reading of nucleotides. 2. Joining the sequences of smaller fragments or contigs together to form a complete genome sequence, and 3. The prediction of promoters and protein coding regions of the genome. Automated identification of genes can be done using the following ways. Number 1. Hidden Markov model or HMM based techniques such as Glimmer and GeneMark. Glimmer, which stands for Gene Locator and Interpolated Markov Model ER, is used to find genes in prokaryotic DNA. It is effective at finding genes in bacteria, archaea, viruses, typically finding 98 to 99% of relatively long protein coding genes. Glimmer was the first system that used the interpolated Markov model to identify coding regions. GeneMark is a generic name for a family of ab initio gene prediction programs developed at the Georgia Institute of Technology in Atlanta. Developed in 1993, original GeneMark was used in 1995 as a primary gene prediction tool for the annotation of the first completely sequenced bacterial genome of Haemophilus influenzae, and in 1996 for the first archaeal genome of Methanococcus janashi. The algorithm introduced in homogeneous 3-periodic Markov chain models of protein coding DNA sequences that became standard in gene prediction as well as Bayesian approach to gene prediction in 2 DNA strands simultaneously. Number 2. Searching the known database of genes such as GenBank to identify genes and the use of algorithms based on decision trees that identify start codons and stop codons of the coding regions. HMM based techniques develop multiple probabilistic state machines, each capable of identifying an ORF. Each machine predicts the next nucleotide character using a state transition with maximum probability and matches the predicted nucleotide character with the current nucleotide character in the actual sequence. Statistical training using known sample sequences derives the probability of state transition. In the case of microbial genomes, the HMM based software such as Glimmer has provided 95 to 97% accuracy. Identifying gene function. After identifying the ORFs or open reading frames, the next step is to annotate the genes with proper structure and function. The function of the gene has been identified using popular sequence search and pairwise gene alignment techniques. The 4 most popular algorithms used for functional annotation of the genes are BLAST and its variations, dynamic programming technique, Smith-Waterman alignment and its variations, indexing based scheme FASTA and its variations, and blocks that uses multiple sequence alignment of conserved domains to identify motifs or characterizing patterns of proteins. Pairwise genome comparison. After the identification of gene functions, a natural step is to perform pairwise genome comparisons. Pairwise genome comparison of a genome against itself provides the details of paralogous genes or duplicated genes that have similar sequence with some variation in function. Pairwise genome comparison of a genome against itself provides the details of paralogous genes, that is, duplicated genes that have similar sequence with some variation in function. Pairwise genome comparisons of a genome against other genomes have been used to identify a wealth of information such as orthologous genes, that is, functionally equivalent genes diverged into 2 genomes due to speciation, different types of gene groups or adjacent genes that are constrained to occur in close proximity due to their involvement in some common higher level function, lateral gene transfer, that is gene transfer from a microorganism that is evolutionarily distinct, gene fusion or gene fission, gene group duplication, gene duplication and difference analysis to identify genes specific to a group of genomes such as pathogens and conserved genes.